looked at this topic of whole genome sequencing in Safefood was back in about 2018 when we had a one-day conference in Dublin, and for lots of the people working in laboratories in Ireland, certainly it was kind of in its infancy at that stage. So I think it's fair to say there have been quite a number of developments since 2018. So it is a good time for us to revisit. The main focus of this morning's is looking at whole genome sequencing, specifically about how it applies in food safety and in detecting foodborne disease. So we do plan to have another webinar, I'll mention that towards the end of this morning's session, that looks more at food industry and how it can be used in the food businesses. So there'll be a little bit of overlap, but today we are specifically looking at the kind of public health, I suppose, aspects of it. And as Tracy said, we have 4 excellent speakers joining us this morning. So I will just give you a quick introduction to the 4. First of all, we'll start with a joint presentation from the Department of Agriculture, Food and the Marine in Ireland. And that will be given by Byrne and Daniel Kelly. Brian holds a PhD in food safety microbiology and is currently working as an agricultural inspector, managing the molecular and foodborne virus section at the Department of Agriculture, Food and the Marine. He serves as the national contact for the National Reference Laboratories for Listeria monocytogenes and coagulase positive staphylococcus, utilizing whole genome sequencing since 2016 to characterize pathogenic bacteria for food safety investigations. And Daniel then works as a laboratory analyst in the molecular and foodborne virus section of the food microbiology division at the Department of Agriculture, Food and the Marine. And his work involves whole genome sequencing of bacterial species such as Listeria monocytogenes, E. coli and Salmonella. And he also supports the operations of Ireland's National Reference Laboratories for Listeria monocytogenes, coagulase positive staphylococcus and foodborne viruses. Then following that presentation, we will hear from Christina Clarke, who is the scientific lead for the Galway Reference Laboratory Service and oversees whole genome sequencing and analysis of these organisms, supporting national surveillance and outbreak investigations. So we're looking at Salmonella there, Listeria. And Christina also serves as the operational contact point with the European Centre for Disease Prevention and Control and collaborates closely with public health authorities in Ireland to enhance the monitoring and control of these pathogens. So Mirko then is our final speaker. Mirko Rossi is a doctor in veterinary medicine with a PhD in epidemiology and control of zoonoses. Since October 2018, he has been working as a scientific officer at the European Food Safety Authority in the Unit of Biological Hazards and Animal Health and Welfare. His main task at EFSA involves supporting the authority in using whole genome sequencing for foodborne pathogens, managing the EFSA data sharing workflow, and supporting the ECDC and European Commission in responding to foodborne outbreaks. So I think we have got the experts in the room for what we want to talk about this morning. So as Tracy said, just to mention, if you have questions, please put them in the Q&A box and then we will go through all of your questions at the end of the 4 presentations. So I will now hand over to Brian, who is going to start off, I think, for DAFM, and I hope you enjoy the presentations. Thank you.
Brian, can I just ask you to unmute? I can't hear you there.
Sorry.
You're great. Thank you very much.
Hello, everybody again. My name is Brian Byrne, and today I'm going to talk to you about the use of whole genome sequencing in food safety investigations. Before I go into the presentation, I'd just like to take this opportunity to thank Safefood for the invitation to chat to you today. And so So just the breakdown or the overview of our presentation. So I'm going to chat to you about our molecular foodborne virus section, and I'm going to go into briefly or talk about whole genome sequencing and what we've done in the department. And followed, I'll go talk about our National Reference Laboratory for Listeria monocytogenes, and give an example of, I suppose, how we work within the food safety investigation process. And Dan will follow up with the laboratory workflow, and also give examples of presentation. So we both work within the molecular and foodborne virus section in the food microbiology division in DAFM. And there on the right is the dream. If you can exclude me, the rest of them are the dream team. We are responsible for we host, our food microbiology hosts the National Reference Laboratory for all the major food pathogens, including Listeria, mono, steak, salmonella, campi, coagulase positive staph, and foodborne viruses. I'm the national contact person for the NRL for Listeria, mono, and coagulase positive staph, and we sequence all these targets. As well as that, we are a core sequencing unit for DAFM. So we also sequence AMR targets and targets that aren't disclosed to food safety, such as bovis, and other bacterial or viral targets that need to be sequenced for the department. I am a data provider for EFSA One Health WGS system, and Mirko will go into detail. It's a very interesting a database that Mirko will go into more detail later on. So today I'm just going to focus on the Listeria monocytogenes, as a reference departure for Listeria monocytogenes. So the whole genome sequencing department started in 2016, and so this came because this UC project started off here. We was a knowledge transfer to incorporate the method into our laboratories. We added the method, we bought the necessary equipment, and in 2020, between 19 and 20, we started validating for accreditation, for the first accreditation in the 17025 accreditation for the Listeria monocytogenes and STEC. And I think we were one of the first laboratories to get accreditation at this time. And then this followed in 2021, we added to our scope by adding coagulase positive staph, salmonella, and AMR targets. And then in 23, we added a further 3 targets: Conibacter, Mycobacterium bovis, and Campylobacter. We also had invested more, and Dan will talk about it later on, in an automated workflow for library prep and DNA extraction. And more recently, in 24, we added a new pipeline to our INAB scope. So who requests WGS analysis from us? So these are the requests, or the institutes we receive requests from. And I suppose the green bubbles are more associated with local or national and European outbreaks or clinical cases, and also the blue bubbles or agencies are associated with maybe source contamination and in some cases research projects. So why we sequence? So there's multiple reasons why the benefits of sequencing. I suppose the most important one, I suppose, is source attribution. So we try to define the source attribution when we're asked for help within a national or international clinical investigation, and also within the scene of a process environment of a food company. We're asked to seek and match the source to contamination of a food product, and we also look for persistence. So is the strain persistent in the environment of the processing plant? And also we have in previous investigations we would ask for resistance profiles. So how resistant are the bacteria or Listeria in this case? How resistant they are to different stresses. And another positive is, I suppose, about whole genome sequencing is the ability to identify false positives. So just example, if a sample comes in from a laboratory and we check against our database, and if it turns out to be an NCTC strain, we can relay this information back to the laboratory that it's actually cross-contamination from the positive control within the process. What is sequence? So the policy and sequencing in the department for Listeria is we sequence all official samples that come into or submitted to the laboratory, but we also try to get the best representative of the profiles in Irish industry. So we try to select isolates from every FBO and different sample matrix within the FBO, and throughout the year as well, try to get the best spread as possible. We also have targeted sequencing from our investigations, that we may target a certain product or food type or whatever the criteria of the investigation is. And on the URL analysis side, Mirko will also, I suppose, speak about this. We also receive requests from a country officer as well on Europe, at the European level investigations. And we take part in research projects as well. So I suppose everyone, what is WGS? So I'm just going to use the car analogy to try to describe what whole genome sequencing is. So if you take a car, or as a Listeria monocytogenes is as the car brand Aston Martin, and it was very expensive. It wouldn't be a car I'd be able to drive or ever able to drive. But and if you take the next level of resolution of the Listeria, it's the stereotype, it's the model of the car. So in this case, it's a DB Aston Martin DB5. And if you go down further into that, into next level of resolution, which is in sequencing terms, is sequence type. And that's characterized as 7 characters within the bacteria that's always present or should be present. And if you put that, suppose, towards a car, you think of 7 parts or components of a car that should be there, such as engine, lights, alloys, or maybe not alloy, wheel, paint, brakes, and so on. So if you got those, the serial number of all those parts and put them in a string, that string of numbers is specific to a particular sequence type. And also in the car analogy, it's specific to just a series type of the car. So if you expand that sequence type of 7 characters to a further 1701 characters, or core genes, that we use core genes to assess a lot of our data. So if you take of, it's a bit like the blueprint of the car. So if you have 1701 characters of car, and that's the same as if you lined up the serial numbers of all those components, and that would be a string, and that you would call that a allelic profile in whole genome sequencing. So just to go, and then the next stage, what happens after that, I suppose, bioinformatics software is used to generate this string of numbers called allelic profile. So this allelic profile is generated, so there's a number assigned for each of the 1701 core loci, and this string of numbers is then compared against our database to see is the same string of numbers in our database. And the way it's done is what's called cluster analysis. So this string of numbers is, when it's compared to, you'll hear the word cluster used a lot today, and thresholds. And basically what that means is, if a sequence that you're testing is between, in this case, 7 or less allelic differences, or has 1694 between 1694 and 1701 characteristics that are similar, they're seen to be the same cluster and they're very genetically similar. So if you see this example, this strain matches with this group of isolates in the database, and you can see they cluster together and they form the same color. This can also lead to the possible source, the detection of the source of contamination. And just another way of graph or visualizing whole genome sequencing is what's called a minimal spanning tree. And so if you think of this as the same data except shown differently, so if you see the bubbles, each bubble is a strain, and there's segments in the bubble. For instance, the environmental, I'm sure you see my mouse, but the environmental and food, there's 3 sections in there. So there's 3 isolates in there, 2 from environment and 1 from food, and they have the identical allelic profile or string of serial numbers. We talk about cars, for example. So the question we had was, what does this target isolate, does this strain that we're testing in our database, does it match that in the database? And this is the isolate we showed earlier. So the same food isolate is found in the same cluster. Sorry, food isolate is found in the same cluster as an environmental food in the same plant. And I suppose the next question is there persistence in the plant? So when you put the sample data that we have for these samples into it, you can see that this isolate was detected in 3 years, over 3 years. And it's the same, it's essentially found within the same cluster. They cluster together, they're very genetically similar. We also look at other genetic, so part of investigations we're asked to look at different other genetic targets. And another, I suppose, is we use the car analogy, is the resistance profile of bacteria. In this case, we look at antimicrobial, more so in the case of Listeria, more so biocide, thermal resistance, and biofilm. Do they produce biofilms? Or do they have genes that are associated with these resistance profiles? And I suppose this is like, in the car analogy, is like airbags, bulletproof glass, safety belts, and so on, that protects the car from anything happening to it. We've also been asked to look for, in certain investigations, at virulence factors. And these virulence factors are, I suppose, will cause harm or cause illness in people. And in the case of James Bond, it causes a lot of harm. So we have other, as I said, this is just a diagram of other analysis we can do other than, so I've spoken about the core genome of 1701. We also can do whole genome, which is a bit like, just it gives an extra bit of a resolution, but we find that there's actually there's never been much difference between the core genome and the whole genome LSD. And in some scenarios, we've been asked to do SNP or SNP analysis as well. So the National Reference Laboratory, as I spoke to before, we hold the reference laboratory for Listeria cytogenese, and in our database, we have two main functions, I suppose. We collect the data from data and isolates, and then we sequence, we molecular characterize a certain portion of those isolates. So just to give you an idea of what we have in our database, and I suppose the inputs into it, we have data or isolates we get submitted from commercial laboratories, and these come from food businesses' own tests. So, for example, a food business has a HACCP plan, risk assess, work assess to look for their Listeria in the process, and they send those samples to the laboratory, and the positive Listeria sample isolates come to us. And on the fishery control laboratory side, we get isolates that are cultured from fishery control samples that are sent to us by the fishery control laboratories. So just to give you an idea of what that looks like, we have approximately 33,000 isolates in our database, and you can see that majority, if not about 97% of the isolates come from commercial submissions. And we roughly receive about 3,500 per annum of Listeria isolates. And on the whole genome sequencing side of things, we have sequenced approximately just a little bit over 17% now. This data was used from August this year. So we have roughly 5,500 sequenced. And you can see from 16, we have grown the sequencing in our laboratory, and we're sequenced roughly 1,000 isolates per annum. And again, if there's a lot of investigations, our sequencing is more targeted, and I suppose it's biased in that regard. Those isolates are not because of the targeted sequencing. And this is the legal basis we use. We use the zoonotic directive to request the isolates and data. We also we have the fish control regulation, and we have ministerial conditions of approval for that supervised plants for meat and dairy. The new regulation that Mirko will definitely be talking about later, I won't go into it, but that's also interesting. As I said, we are a data provider for the EFSA One Health database. And at the moment, our policy is we upload our official isolates to the database. And this will change during investigations because of this new regulation next year with that. All isolates that are associated with outbreak investigation will be uploaded to the database. So this is just to give you kind of an idea of how this process works. We receive a file or a WGS file from our colleagues. In some cases, it's Christina who's going to speak next. And we get the request, we are asked to look into or compare this profile against our database. And if we find that there's no match, or this profile is unique and it's not clustering, as in there's not within 7 allelic difference, as I spoke to before. We would report back to the competent authority and the requester that there's no match in our database. And if we have a match, we report the data, say we send the matching data and information on the, if we have information on FBO, we send it to the competent authority that's managing the investigation. But also it's important that the competent authority that manages the investigation use both the WGS data and their own epidemiological data obtained from their investigation to come to make the decision on the potential source of the contamination. So in summary, the NRL, we collect and characterize strains. Now I've focused on Listeria, where we sequence all pathogens in a laboratory. We've helped with local and European investigations. We've also worked with our competent authorities in working with FBOs to try to find try to resolve the problem they may have. But I suppose, as I just said before, the most important thing with whole genome sequencing is to use it not solely as the source of evidence, and use it with epidemiological data when making the decision. So I'd just like to say thank you for your attention, and I'm going to pass you on to Dan. Dan is going to share his presentation now. And
thank you very much for that, Brian. Hello, everybody. I'm just going to share my screen very quickly. So, yeah, thank you very much for that, Brian, for your excellent overview of the WGS section. Daniel Kelly is my name. I'm a laboratory analyst in the Whole Genome Sequencing Laboratory. So today I would just like to follow up with Brian's presentation and just give a very brief overview of the WGS laboratory workflow, and also how that might be applied in an investigation situation. So as Brian mentioned, we receive bacterial isolates from various commercial and private laboratories and official laboratories nationally. So the first step of the WGS workflow will be to culture these bacterial isolates. We would primarily do this by culturing the bacteria on a general purpose growth medium for 24 hours at 37 degrees. As you can see, I have just pictured here what that might look like after 24 hours on a Columbia agar plate. Once this is achieved, we can then extract the DNA from the bacteria. And in our laboratory, we have two separate methods of doing this. Primarily, we would use an automated workflow. In our laboratory, we have a Roche MagnaPure robotic workstation, and you can see myself pictured there hard at work using this instrument. This is a high throughput instrument. We can extract up to 96 bacterial isolates at one time. If this isn't applicable, we can also use a manual extraction method to perform our DNA extraction. Either way, we would also assess the quality and quantity of the DNA prior to progressing to DNA library prep. So in our laboratory, we use the Illumina sequencing platform. So the next step will be to prepare the DNA libraries using the Illumina DNA prep workflow. So in a nutshell, this is essentially converting the extracted DNA into a sequencing ready DNA libraries that the sequencers can read and sequence. Similarly to the DNA extraction, we can use an automated workflow, and primarily we use the Hamilton Star automated liquid handler, which you can see one of my colleagues pictured, again, hard at work. This workflow takes about 6 hours to complete. We can also do this manually as well, but depending on the number of samples, this might take as long as 2 days to complete. So we definitely prefer the automated workflow. Once this is achieved, we can then move to the sequencing stage. As I mentioned, sequencing is performed using the Illumina platform. In our laboratory, we have 3 sequencers, NextSeq 1000, NextSeq 2000, and a MySeq. So depending on the number of species and the type of species, we can use any one of these instruments. And what these do is essentially generate sequencing data as a FASTQ file output, which we can use then for our bioinformatic analysis. So we would use Ride-on SeqSphere bioinformatics software primarily, well, firstly to check the quality of the sequencing data. And once we're satisfied with that, we can then generate outputs that Brian mentioned earlier on, such as a CG-MLST profile, and also looking at the virulence and resistance profile as well. So what would that look like then in a real world situation? So just to give a brief example, an official laboratory submitted Listeria monocytogenes typing request to our laboratory in relation to a suspected food alert for a specific FBO. So 10 isolates were submitted to the laboratory with two primary queries. Firstly, how related these isolates are to one another, and secondly, how related these isolates are to previously sequenced bacterial isolates from this specific FBO, and also if there's any evidence of a persistent strain. So performing the CG-MLST analysis that Brian mentioned a little bit earlier, we were able to generate a minimum spanning tree of the 10 isolates. And you can see here pictured the 10 isolates where one allele in difference or less. As you can see, each one of these circles represents a bacterial isolate. So we could see straight away that they were very much related. So for the second question, we had to identify if there was any existing isolates in our database. We were able to do this by looking at our metadata and identifying any previously sequenced isolates for this particular FBO, and we were able to find 19 of these isolates. So when we performed another CG-MLST, including these 19 isolates and the original 10 official laboratory isolates, we could also again see that there was less than 7 allelic differences among these isolates, so we could say that they were indeed related. Using this metadata as well, we were able to identify that there was both environmental and food samples involved in this strain, and also that the same strain was detected in the FBO in 2019, 2020, and 2021. So it was indeed a persistent strain. So this really highlighted to us that although the sequencing data was important, the metadata used in context with this really was where the power was. So finally, I just want to say thank you very much, not only to Brian for allowing me to present today with him, but also for the rest of the team. So thank you very much, guys. That's the end of our presentation. So I'm going to just stop sharing my screen, and I will pass you over to Christina, who's our next presenter. Thank you very much.
Thank you very much, Dan, for your lovely presentation. You guys went through the core genome MLST very nicely there, so I think I can go through that quite quickly here. So my name is Christina Clarke. I'm the Specialist Medical Scientist and Scientific Lead of Galway Reference Laboratory Service. For today, I'm going to mostly focus on Salmonella and Listeria, but all the organisms, we cover other organisms. So who are we? We're a Galway Reference Laboratory Service, which is comprised of both the National Salmonella, Shigella and Listeria Reference Laboratory for human isolates and the National CPE Reference Laboratory for human isolates. And we're based in the microbiology department on the campus of University Hospital Galway. And what do we do? So we do whole genome sequencing and analysis of all human Salmonella, Shigella, and Listeria isolates. Unfortunately, we can only accept isolates. Sometimes these organisms are non-retrievable from the human sample. This can be for a number of reasons, like previous antibiotic therapy, and occasionally if they're at the tail end of an infection as well, the laboratories just can't isolate it out. We also sequence all CP isolates from diagnostic samples. So that would be like your bloods, your urines, wounds, things like that. But we also sequence a large proportion of screening samples. So that would be like your rectal swabs or feces. We also do a limited number of carbapenem resistant Acinetobacter baumannii and carbapenem-resistant Pseudomonas aeruginosa isolates. So all these activities add up to approximately 2,000 CPE or CPO isolates, about 140 Shigella isolates, about 400 Salmonellas, and 10 to 20 Listerias. Thankfully, the Listeria burden in humans tends to be quite low in Ireland. We only really see samples from patients who have infections in their blood or their CSF, and that tends to be people who are either at the extremes of life, very elderly or very young, and we would also see this with pregnant people as well. So what else do? We support clinical laboratories by confirming and subtyping their Salmonella, Shigella, Listeria, and CP isolates. We provide national data to inform public health on the scope of issues, outbreaks, and the effectiveness of responses. We also liaise with the HPSC and the European Centre for Disease Control to aid excuse me, to help identify and control international dimensions to outbreaks. We also liaise with the Department of Agriculture and the Marine, so that would be Brian and Dan, for any of our Listeria outbreaks to help identify and help control national outbreaks. This involves the sharing of human sequencing data without any patient identifying data. And this information helps guide public health intervention and control transmission of infection. So if we see like a large cluster, and we go back to DAF and they have a match, oftentimes then an outbreak management team could convene and help try and reduce the numbers. I thought I'd go through some of the trends we've seen in Salmonella, Shigella, and Listeria as well. So you can see a big dip around 2020 and 2021 for our Salmonellas, and that's because about 50% of our Salmonellas are travel acquired, and the rest then tend to be homegrown Salmonellas. So for our non-typhoidal Salmonellas in 2024, we actually saw 412. We did see 130 cases of Shigella, 39 cases of typhoid Salmonella, which were all travel related. And for Listeria, it was our highest number in quite a few years at 21. Whilst the Listeria numbers are quite low, it does have significant morbidity and mortality for patients. That's why we like to make sure we sequence everything that we can for them. So the top 10 serotypes of non-typhoidal Salmonella that were submitted in 2024 and 2023. So you can see enteritidis tends to be our number one year on year, followed by typhimurium. Then we have our monophasic typhimurium. And after that, it can vary year to year. This just gives a distribution of the different serotypes in 2024. So you can see the others make up a large proportion of it. And then our Salmonella typhus and paratyphus. So our typhi, S. typhi is the most common one, followed by paratyphi A and then paratyphi B. And this seems to be the same year on year. There are a number of public health strategies trying to promote people to get the Salmonella typhi vaccine if they're traveling to any endemic areas, because it's quite effective. So also, antimicrobial resistance is increasing year on year as well. So this table just gives you an idea of the percentage resistance in the various serotypes. And this is all from 2024 as well. So you can see Salmonella enteritidis, about a quarter of those would be resistant to Cipro, and about 8 to 10% would be resistant to amoxicillin. And those would be kind of common enough antibiotics. So that can be worrying. So typhimurium, then we have a larger percentage that would be resistant to amoxicillin at about 22%. Salmonella typhi, we are seeing a lot of MDR and XDR typhus, so they can be challenging to treat. For our Listeria serotypes, predicted from 2020 to 2024, the most common one that we see used to be always 4B, but now it seems to be 12A. As you can see, the numbers in 2020 and 21 were quite low, even though most of our Listerias tend to be acquired in Ireland. They're not quite sure why those years that they were so much lower, because you wouldn't expect when it's not usually travel related. So what technology do we use? So we use a different DNA extraction method than the food laboratories. We use a smaller instrument. It does about 24 at a time. So it's the DNA extraction unit called QIAGEN Easy2 Connect MDX. So it's a cartridge-based system that utilizes magnetic beads for the purification of nucleic acids. These cartridges are single use, and you can probably see that there's all separate lanes for each cartridge. So each cartridge is single use, the pipettes are single use. This helps us to avoid contamination, which we find especially important. We also have a robot for liquid handling. We have an older instrument called a Biomic 4000, and we also use the Illumina DNA prep kit that takes us roughly the 6 hours as well to perform it with our instrument. So this does all our library preparation for our sequencing runs. We would do our quality checks at the end of the library prep to make sure that the DNA quality is good enough and that the quantity is there as well. We'd also look at determine the fragment size to make sure that it's appropriate for sequencing. We also use Illumina NextSeq 1000, and we use the XLEAP P1600 cycle kit. So that gives us all the information we need. This sequencing takes about 34 hours, which is much improved on our old instrument. Our old instrument was the MySeq, and it would take about 56 hours. So we save a full day using the NextSeq, as well as having a much higher throughput. So for analysis, we use the same bioinformatic pipeline. So it's the Bruker BioSeq Random Typer. I think they only changed their name in the last couple of weeks. So this would do our quality trimming and our assembly. For our de novo assembly, we would use either Skiza or Spades. For quality scoring, we would look at FastQC, N50 coverage, contamination check using MASH. We would do our genoserotyping or serogrouping, MLST and CG MLST analysis. So the genotyping, the genoserotyping serogrouping is useful. We don't actually do any serology anymore in the reference laboratory. We do all genoserotyping. We also would look at AMR detection using the NCBI AMR Finder Plus, and this looks for both acquired resistance and point mutations. So for our cluster designation, basically all isolates are analyzed together with those of either the same clonal complex, sequence type, or MLST type. Once they're found to be closely related, are given a cluster designation. And when an isolate is identified as part of the cluster is designated an alphanumeric string in which the first 1 or 2 characters are related to the species. The first 2 numerals indicate the year, and the final 3 numbers indicate the order of identification. So for example, S24003 is the 3rd cluster of Salmonella and Listeria identified in 2024. So unfortunately, I couldn't share any real outbreaks at the moment. They're due for publication, but I did do a simulation of what an international cluster would look like. So basically, this is an MST. These are generated every time we do a sequence, every time we look at it, every single isolate. So, for example, we would see an EpiPulse event pop up on their website. We would download their sequence and we would compare it to our own database. So this simulated EpiPulse event happened in 2023. And you can see that we had 2 identical ones because they're all part of the same circle right here. So that's a 3-part, almost like a pie. So that we had 2 identical matches, and then we also had others related in 2023 to it. 2024 and 2025, this would be consistent where there was a reservoir in a particular country. So we'll say EpiPulse event was created when country A found that citizens were coming home with infections post-travel. They wanted to know if other people are seeing it too. In this case, so we queried it on our database, found the matches, we got back to the HPSC. So we would report our microbiological findings on EpiPulse, and the HPSC would report their epidemiological findings. And for example, they could say that all the people traveled to the same country. They may have been in the same, if it's a very large country, they might say the particular regions. Then that would also get, if it was found to be a specific region, that query might go to the food EFSA people, and they would see if they had any food matches. But in a situation like this, where we could see cases returning year on year, which we have seen in a few instances. So once there is that reservoir there, sometimes it can be seasonal, for example, related to certain produce. You'll have to excuse me, I woke up with a bit of a sore throat this morning. And this would be a simulation of a domestic cluster. So simulated human cases are in pink, and the simulated DAFN cases are in blue. So this is a simulated outbreak that could have been seen over 2 years. So when a human cluster arises with no obvious travel history, the HPSC often asks us to share our sequences with DAFN. If they find a match in their database, then the relevant authorities are informed, and sometimes an outbreak management team would be assembled. Sometimes this will lead to different remedial measures, whether it be a food item being taken off the shelves, or if some investigations need to happen at a farm level. I wouldn't have much info on what happens on the food side. So for our surveillance, both national and international, so for national surveillance, each Salmonella, Shigella, and Listeria isolate is reported to CIDR, but there would also be a corresponding CIDR event from the hospital where the isolate first came from. Each new cluster is reported to the HPSC. Additions to clusters are reported to the HPSC. The HPSC then liaise with their local public health, and there could be like trawling questionnaires issued to patients to see where they were, what they've eaten, different things like that. And an EpiPulse event may be created if epidemiology is suggestive that this may be an international or travel-related cluster. So for national surveillance, concerning clusters may warrant further investigation or declaring an outbreak. We liaise with our DAFN colleagues when requested by the HPSC and determine if they have related isolates that outbreak management team might meet, decide on further measures. And always any isolates that are from a suspected outbreak, we would prioritize them for WGS. While we do sequence all Salmonella, Shigella, Listeria isolates anyways, anything from a suspected outbreak will jump the queue to get the result out as quick as possible. So as well, I send monthly line listings to the HPSC, public health, hospitals, and Safefood as well, so that everyone can see what the trends are like with those pathogens. The AMR data is sent quarterly for Salmonella and Shigella to the HPSC, and they release data regularly via their quarterly reports and annual reports, which are available on their website. Both quarterly and annual reports share data on AMR amongst the Salmonella and Shigella. We also release annual reports on our activities, which are available on, it's still the CLT website, but it will be changing to the HSE West, Northwest website. So then for more on the international side of things, we use the ECDC EpiPulse system for Salmonella, Shigella, Listeria, and we actually use it for the healthcare associated infections. So basically, it's an online portal for European public health authorities and other global partners. It's used to collect, analyze, share, discuss data for threat detection, monitoring, risk assessment, and outbreak response. It's an indicator-based and surveillance-based system. So we compare the all European outbreak sequences or any EpiPulse events, which sometimes can come from further afield, like the US, Canada. We respond to all EpiPulse events. We've informed the HPSC of any matching isolates. We're also real-time reporting our WGS data from all Irish isolates to the CDC EpiPulse cases system. This is kind of what I feel that it's better to move towards this, because we can see signals much sooner if there is an international element to outbreaks. So different challenges we've had over the years. And currently we have no electronic reporting facility. The actual buildings itself on the campus at UH are quite old, and we do have some difficulty in sharing sequences. Hopefully with some of the upgrades that are due within the HSE, we'll have electronic reporting. And we've been promised a new lab for a number of years, so hopefully that will happen soon. But we have also overcome challenges in the last few years. So we used to have a lot of WGS and analysis delays. This was due to, well, older machinery such as our MySeqs. We had a lot of difficulty with those over the years. But now that we've upgraded to the NextSeq, oh, thank goodness it seems to be working well for us. We also have semi-automated our analysis. So previously we had to analyze things more manually. So we'd come in on a Monday after the sequencing was done, and it would take a good 24 hours before everything was analyzed. Now we just come in on a Monday, we can report all our results. So our turnaround times has vastly improved. It used to be 28 working days, our turnaround time, but most of our salmonellas are out within 10. And the quickest we've ever had out is about 3 days. So thank you for listening. And I just want to acknowledge all the team in the Galway Reference Laboratory Service. So that Alma, Ger, Paula, Fina, and Prof. Cormacan, they're all a wonderful bunch to work with, and what we do wouldn't be possible without all their help. Thank you very much. And I will pass you on to Mirko.
Hello, thank you. Do you hear me very well? I hope yes. Okay, so I'm sharing my screen. Okay. Thanks again for inviting us to make this presentation in this forum. We are very, very grateful for this invitation. So today I'm going to show you announcing One Health in the European Union through the WGS, One Health WGS system. Before we move to that topic, just a very few slides on what is EFSA, for who doesn't know. And so EFSA have been established by the European law in 2002 after following a series of food crises. The objective was to improve the food system at European level, to restore and maintain the confidence of the food supply system at European level, to help ensuring high level of consumer protection, and this is very important, to clearly separate risk management action function from risk assessment. We are seated in Parma, in Italy. What we do is provide independent scientific advice and support to European Union risk managers and to policymakers on the food and fish safety. We provide independent timely risk communication and promote scientific cooperation. What we does not do is to develop food safety policy and legislation. We do not adopt regulation authorizing marketing new products, and we don't enforce food safety legislation. Within the scope of the EFSA remit, in 2019, the European Commission have been mandated, EFSA and also ECDC, to establish a new platform, which have been the scope to collect WGS data from each remit and share it in a joint fashion to support outbreak detection and investigation at the European level. We have been taking consideration a lot of critical points during implementation to ensure secure collaboration, to be sure about responsible data use, taking into consideration the EU complexity, but maintaining an implementation that is operational efficient and could constantly evolve. These are the prerequisites of our implementation. So the system is composed by two platforms, independent platform, one sitting in CDC, one sitting in EFSA. Why we have two platforms? There are several reasons for that. First of all, we need to address sector-specific governance and agency network needs. We have to streamline user identity management and user support. Being two different organizations, we have different type of user, different agency-specific infrastructure to take into consideration. And finally, we also need in both sides to control, having a higher control of the data sharing protocol from data on food sector toward public health and vice versa. The two platforms are interoperable. They can easily exchange with the machine-to-machine protocol relevant WGS-based type information and epidemiological data upon a verified correlation. So just to say, if it's the Look2 platform, they are one single system because when relevant, the data is always shared. Focus on the EFSA platform of the One Health WGS system. The goals are on different domains. First, in preparedness, we need to build a database of genomic profiles that can be queried in case of woven outbreaks. So, very similar to what have been presented to you by the Irish colleague is happening also at European level. But we need also to have a system that is able to activate the correct response, supporting a real-time investigation of multi-country foodborne outbreaks. And for doing that, we need also to ensure a good engagement within sector, among member states of the food sector, but also and especially cross-sector. And finally, we need also to have a system that gives the opportunity to be compliant to the regulations, which is particularly important from next year. The data are shared basically on voluntary basis at the moment. Starting from 23rd of August 2026, there is also a mandatory submission which fall in scope of the Commission Implementing Regulation 2025-179. But during voluntary activity, the member states are usually choose two main approach. One is a reactively data sharing, which is, I would say, currently the majority covering the majority of the cases of data sharing in our system, which is data calls from all countries when an actual outbreak assessment has been initiated. You might be aware that EFSA collaborate with the CDC, and the ECDC and the European Commission request in case of multi-country foodborne outbreaks to participate in a common joint assessment, and this is happening through what we call it rapid outbreak assessment publications. When this is initiated, EFSA always open a data call to all countries, and this is a moment where the majority of the data or majority of the country participate in data sharing to EFSA. However, there are every year a larger number of countries that are starting to do a proactively data sharing. So share the data, regardless if there is actually an actual ongoing outbreak. Of course, the strategy of what to share have been discussed in several network meetings, but it depends on the policy and of each country and the strategy they want to use it. Some countries are systematically share our official control data they are collecting, other countries no. Going more specifically on Commission Implementing Regulation, the new regulation that will be in place, it is already published, and you can find in the journal, official journal of the European Commission, or European Union, but it's going to enter in force starting from next year in August. I include a QR code on the right for you to go immediately to a document published together with the law by the European Commission to solve a lot of questions that have been raised during the drafting of the legislation. In a nutshell, the purpose of the legislator is to mandate WGS data production and transmission to EFSA of those isolated in the food, feed, animal, and related environment, which are associated or suspected to be associated to foodborne outbreaks. The pathogen cover are Salmonella enterica, Listeria monocytogenes, E. coli, specifically STEC, Campylobacter jejuni, and Campylobacter coli. There are specific requirements about accreditation and also about the fact that the transmission should be happen without a new delay, which means as soon as the data is available. And there are a set of indication of additional metadata that need to be transferred to EFSA when there is an obligation to perform this data transmission. There is also a part of the article referring to the participation of the food business operator, which is they must cooperate with the National Food Authority in case of food outbreak and when relevant, share data. But let's go back on actual system. It is a closed system. It's not open to the public. So the user who actually can access the system are defined at member states, are appointed. And this is this control access ensure the maintenance of the chain of custody information we are collecting, which for EFSA is very, very important to always go back on the chain and identify the correct person who actually takes the responsibility of the specific data transmission. The PAF committee have been identified that the food safety authority for each country should identify a country officer who is coordinating the data provider organization and be a contact point for EFSA and has the visibility of the entire country transmission effort. And the country officer is a person who interacting with EFSA appoint each data provider. Each data provider submit the data on behalf of their organization and have also visibility of the organization data transmission effort. Both the country officer and data provider can also explore ECDC database of human by a query capacity. In this slide, I try to explain you what we actually store in our system. And what we have divided the storage in two main data type. One is the dark green is the WGS data, which is composed by information about the experiment. So information about how the raw sequencing data have been generating, and the actual derivation from that data of typing information, including the genomic profile. Our system and ECDC system are based on CG and ST profile, but also other information such as serotype determination, AMR profiles, valence profiles. Also, we are collecting epidemiological data, and this is just information related to the sample from which the isolate have been collected. And what is important is an asynchronous is asynchron. So the two data type might be owned by different data owners. Therefore, it's possible in our system to asynchronously submit first the genomic data and in a second moment the epi data. And this is just because we want to ensure the complexity of the European Union, which is sometimes different data is owned by different organizations, different users. In addition, all the time the data in our system is controlled by the data providers. So the data provider is the main owner of the data, and they can always control the status if the data is available to be shared to other users or needs to be withheld and unreleased from the database. We have developed multiple ways for interacting with us. And this is also for addressing all complexity at European level in advance in the use of WGS system, WGS technology. So we have developing a user interface, which we call WGS portal, which provide unsupervised and seamless interaction for country needing full support. So this type is supervised, non-unsupervised. It provides supervised and seamless interaction. So in practice, the member state transmit raw sequencing read with the portal to our system using our computing resources to generating the profiles and the other type of information, and then release that information to our database. For other type of member states who have much more a different type of implementation, the analysis which is happening in our system can be transferred in the local, and through a machine-to-machine interaction and the consumption of API, dedicated API, they can directly transmit the data without the need to share or to transmit raw sequencing data. But we are constantly evolving and improving our system. We have expanding the scope, and this is going to be happening in summer 2026. We expanding our data scope. We are going to accept also assembly. We make a clear separation between monitoring data flow and mandatory data flow. We are going to enhance metadata to meet regulatory requirement, including Campylobacter, ensuring robust data transmission by allowing redundancy. So everything activity that can be done through the UI can be also repeated in API. And we have established new roles, like specific role, like something like a role of person that can submit on behalf of that organization in Europe, and also specific situation like Northern Ireland user who has specific interaction linked to the withdrawal from the European Union. We have a change management plan with webinars and training, so stay tuned. In early 2026, we come back to all our stakeholders with a specific plan. But let's go inside of the current situation. Our database is composed currently over around 18,000, something like that. Today, this data I'm showing is a data of a few weeks ago. We have divided in two parts. On the left side is the actual contributor of the member states with the submission, which we call it private, so direct submission to our system. Total around 5,000 submission from 29 countries. But as you can see on the bottom, we have a huge bias towards certain countries that consistently represent over 70% of our database versus very little contribution from other countries. We can analyze in the different level of contribution by different reason, by the size of the country, by the fact that the WGS is not well used still in some of the country. But also, some of these countries are actually countries that very use or WGS, but do not provide a systematic sharing with EFSA on the data. One very important is that among these almost 5,000 data that have been available to CDC to query in the last 3 years, a little bit over half of that actually are found matched with this CDC cluster. But in order to support the CDC work, we also populate the data with public available data automatically. We are retrieving since March 2025, automatically import from NCBI data that have been submitted in the scope as food or environmental issues. So far, we're collecting around 12,000 data, and of which 32% are matching the CDC cluster. One very important note that the majority, as you might know, 3/4 of the data submitted are mainly US, UK, as you probably know, and very little is from European Union member states. Meaning that these 5,000 data actually are most likely the most populated international EU-based food data that we currently have. But how we use the data? The data is used solely for performing joint rapid outbreak assessment. So the data being published only in case there is a joint data outbreak assessment, but it's used daily in our monitoring activity with the CDC. The CDC are meeting every Thursday afternoon at 1 o'clock approximately. We discuss of all possible cluster and all possible event opening pools and cluster detected in our system. And for performing this action, CDC is querying EFSA recurrently on a weekly basis using a fixed threshold for getting possible matches. And during a specific event, CDC is also able to query our system using a variable threshold as needed. We perform automatic data share exchange. We don't have a human supervision data based on rule of engagement, and that allows us to always perform cross-sectorial analysis. This is just in a nutshell what we exchange. We always exchange type information and only the type information. So we never exchange the full sequence or assembly. We change the profile and other typing data. We also change always very minimum metadata, in particular the full category and the sampling year, but we not always share the country. This is on the shoulder of the data provider who can decide to hide or share country information to ECDC. Public health users. When they decide to share, then all country information, country of sampling, and country of submitting orientation, then being shared. But just very briefly, technology-wise, how this integration works. So as I said, our system is designed on CGMST profile. What is designed is also what we call serverless CGMST allele designation. So you know that what is CGMST? Is a transformation of a sequence of one gene. For example, this is a sequence of locus A. And by integrating a specific nomenclature server, we assign a value of that sequence. We call it 1. But if the same sequence is pushing within a different server, nomenclature server, the value of this allele designation might change because it is the list of this server 2 might be different. For example, in this example, the same identical sequence assigned 1 in the first example, assigned 23 in the second example. By by eliminating the need of a magnitude server, what we can do, we can transform the sequence in a number by using a function, so an hash function, so a transformation, a computational transformation. This is just a number, as is a number in other situation, but is a unique number. So this function will always transform this sequence in this number. That is allowing a unique profiling. What is the cons and pro? The cons, there are a few. There have been possible what we call collisions, meaning that different sequence might having the same designation. And most important, we lose the possibility to create the list of our schema by allowing potentially spoiling the list. But we have much more pro, like we have seamless integrations of cross-sector, the possibility to generate the sequence, the impossibility to go back to sequence by the hash function, we don't need anymore to maintain a manager server, and we don't need to access the same manager server. And this will allow us to be fully integrated with the CDC. So by exchanging all the CGM and ST profile. This is just an example how query works. They perform a cluster analysis. They collect all the elements belong to this cluster, submit to us, and ask, can you give me something at the n nearest distance of any of these profiles? And our automatically calculate this and reply to CDC automatically by giving the entries at a range of distance as supposedly designed in our collaboration agreement. In summary, this system now contains day by day increasing number of sequencing, over 1,700 for the EFSA data, over 17,000 profile for EFSA, over 20,000 from CDC. We still have a lot of issue in unbiased data, because we still, both in the CDC EFSA, we still have a lot of data for few countries and less data for others, but we are working to improve that. This system have been hundreds of thousands of interactions between the two platforms. We are speaking today about 285,000 interaction queries between the two systems in the last 3 years without any drawbacks or any problem. We identify over 300 clusters, and which were used for assess 10 major outbreaks. EFSA is constantly engaged in announcing activity through interacting with stakeholders at European level, national level, through performing initiatives such as increase awareness of risk management policymaker, especially with the Science Meet Policy Conference, that is, you can find it in this link. And stay tuned, because we'll be probably come up as another Science Meet Policy next September, and a continued announcement of the WGS system. I've finished with this slide. There is a topic page dedicated on our website that you can identify with our QR code, and a promotional video for enlarging the awareness of the use of WGS system in our setting. For any question, you can always contact me and submit and ask a question to our system. Thank you.
Thanks, Mirko. And we are going to progress to the question and answer session, which Linda will begin with.
Okay, great. Thank you. And thank you to the 4 speakers for their excellent presentations. And we have a number of questions in the Q&A box. So please add your questions. There's still time to do that. But in the meantime, we'll get started. And the first question is, and it's not directed at any particular speaker, so feel free to chime in. Are EpiPulse and EpiS the same thing? So I don't know if anybody wants to.
Yeah. I think they used to be called that, but we used to call them EPIs, even though EpiPulse cases is the.
Yeah, I can confirm, if I can jump. Sorry, Christina.
Yes.
EPIs was the old version. Now they make a refactoring last year, 2 years ago actually. So now it's called EpiPulse, and there have been division between EpiPulse events or threats, that actually is what Christina was mentioned, and EpiPulse case, which is their system at the CDC, where they actually collect indicator-based surveillance from the European Union Public Health Authority. So EpiPulse is actually the new one-shot place for ECDC data collection.
Perfect, thank you. So there's another question now here. How does a 7 allele difference used in the Republic of Ireland compare to a 5 SNP cluster used by authorities in the UK? Are they the same thing or are they comparable? So I don't know who wants to talk about the differences between Ireland and UK.
I suppose,
Right.
Can you hear me okay?
Yeah.
Yep. So I suppose it's the threshold is used, different thresholds are used for different pathogens. And just like what Mirko was saying, that we could produce a sequence from a particular schema, or in our case, we use the same as Christina and also our colleagues in Cherry Orchard. And then we get a file from the UK, we bring that into our scheme to compare, rather than to compare the two SNP and Whole Genome MSD. So when during our validation for Listeria, we wouldn't really use SNP too often. For other targets, we have used it in the past, but that's what the comparison is taken away in a way. Is the variables are reduced by taking the raw data file and bringing it into our system.
Okay. Thanks, Brian, for that. And so now a question on how do you see machine learning or AI supporting WGS-based pathogen surveillance? So this is a bit of a look ahead. So does anybody want to talk about AI in whole genome sequencing?
Maybe if I can join that.
Yes.
Currently, there are some initiative on especially using machine learning for the prediction of, for example, AMR phenotype through the nice WGS sequence information. This is a classical use case of AI, or more precisely, machine learning. There have been also some impact on machine learning again, not properly what we know as AI now, but more on that side of the AI world, about in a certain case use CGMST data or SNP analysis for improve source attribution in a certain condition, or for the identification of possible characteristic that might be relevant, for example, in Listeria surveillance in environmental production environment or other type of information. This is a more or less current situation about AI use based on my knowledge.
Great, thank you, Mirko. And the next question is, do you see WGS becoming a standard requirement for all foodborne pathogen testing worldwide? So, don't know if anyone wants to tackle that one. Predictions, Gayer.
I can take eventually again,
Yes,
but I don't
okay.
want to overcome the others, so please to stop me. But this is very long discussion, honestly. So we had in pre-Trump time a long interaction with the FDA, for example, and American CDC on this actual topic, with the European Union and other member states, and it's more like it's not a sort of obligation, but it's becoming a sort of a scientific obligation. So we are getting rid of what is in the past was the main technicality for detecting typing or for performing typing. So we are becoming more dependent to WGS data for performing even simple information such as extracting serotype from Salmonella. We know, for example, a lot of laboratories in EU, they are stopping to make serotyping by classical methodology, so they are relying on WGS data. So we are taking the advantage of WGS data, but also we are becoming dependent to that. So I would say that is a sort of interconnection of these kind of situations. But I will leave other colleagues to answer eventually.
Does anybody else want to comment? Brian, do you want to come in there?
Yeah, just to add to what Mirko was saying, so as part of we're the NRL, the Irish NRL, where we have URL workshops annually, and we discuss whole genome sequencing. There's also a working group at the European level where each European reference laboratory takes a pillar of whole genome sequencing and has developed that over the number of years, and it's been very good. Because it's the same technology, essentially, same method, but So there's training and different pillars within the working group. But within our Listeria URL workshop, like what Mirko was saying, that people are some member states are they're not using it, and but there's more after coming on in the last few years. But there's also resources as well. People or some laboratories may not have the resources to do all pathogens for sequencing, and they may also, some others contract out sequencing as well. So there's an awful lot more laboratories doing sequencing now, and yeah, it has come on, but there's still a few member states anyway that haven't started doing or putting as much resource into it.
Okay, great. Thank you for that. We are okay to move on to the next question. So how has whole genome sequencing changed how we detect and respond to foodborne outbreaks compared to older typing methods? Anyone want to take that one? Christina, do you want to unmute yourself there?
I suppose from a human perspective, at least now, when it was just serotyping, there was a lot more guessing and just MLVA. At least with WGS, we were able to say that, yes, these patients have the same exact Salmonella, try and find out where the source is from there, and that we can query with our colleagues over in DAFM. Whereas previous to WGS, well, there was a lot more guesswork, I suppose.
Okay, thank you. Anybody else? Brian, do you want to comment?
Yeah, just add what Christina was saying. We find we're given we're given a lot of information, where beforehand we may have got a positive on the food industry side of things. We get a positive in a particular FBO, and then there's a lot of it takes I suppose you presume that environmental, for example, could be the cause of contamination. So because it's so accurately defines a particular profile, it adds that extra layer of resolution that you don't have with CCMSTs or serotype that you get in other targets. And I think in FBOs, and I always find it interesting when I hear the story coming back from the competent authorities, because it links with the whole genome sequencing data, and you can see it can help, I suppose, identify a source to a particular strain in the plant, and it in some cases gives you too much information. It makes it more complicated to make a decision, but the level of resolution is great for the purpose of trying to detect the source.
Perfect, thanks, Brian. More questions coming in there. This is one that's for Mirko. A colleague from FSA in Northern Ireland is asking, who should we link up with at EFSA or the EC to discuss linking into WGS portal and complying with 2025-179?
Yeah, I will invite the colleague to contact Tracy or you, Linda, that you have been instructed with my mail, and then to bilateral we will answer the situation. Yes.
Okay. So we can put them in touch with you directly, and you can take it from there.
And then I can forward the interaction with Sante. So in particular, Sante G5, Director G5 is the one oversee the regulation implementation, and us from the data transmission. So for answering the question how much mandatory will be, so the obligation of Northern Ireland should be request to comment from G5 to the actual technicality how to access our system, just to drop an email to me and we will update the colleagues.
Great, thank you so much. The recording will be available, so I suppose people will see the slides. Tracy, am I correct? You might just...
Yes, correct. Just because presentations are basically intellectual property, we prefer not to offer slides. We'd rather you use the presentation that's recorded. So it will be shared in the next couple of days.
Okay, great, thank you. And so, okay, another question on how quickly can EFSA and ECDC produce a joint assessment once an outbreak signal is detected? So,
Okay. So I can tell you that from the moment the CDC or the European Commission requests the production of the rapid outbreak assessment, we have a maximum of 6 weeks to produce the assessment, which is include at least 1 week for data collection and 1 week for consultation of all stakeholders for data correction. We are discussing now with the Commission and CDC to shorten in a certain condition with certain type of data from 6 weeks to 4 days, 4 working days. In a certain condition, of course, the quality of the data will be changed. But the question is more specific, and this is a little bit more difficult. So when the outbreak signal is detected, then and time with making assessment might be vary a lot because it's depending on the requester, which is CDC or Commission. I make an example. We monitoring every year 100 outbreaks, more or less. Every week, 16 to 20 outbreaks we are discussing in our weekly meeting. Some outbreak we are just having, I say, specific attention because of the size or the impact of the multi-country dimension, etc. Sometimes we don't get any requests. Sometimes we get requests. It's really depending on prioritization at the CDC Commission side and also what the countries want to do, because sometimes there is not a request from the member state to perform a EU assessment. So it's really depending on the situation. Is it ad hoc all the time from the signal? But I can be more precise when actually there is a request.
Okay, great, thank you. The next question is, does the National Reference Lab notify the relevant competent authority if isolates are identified in an FBO commercial sample?
Yeah, so when we receive isolates, we don't notify this. It's responsibility for the FBO to notify the competent authority if there's an issue. So if we might be contacted by a competent authority, or another NRL with requests. So in the case of a commercial sample, if a competent authority is part of they're doing an investigation on a particular food business, that's the only we discuss the whole genome sequencing profiles then, but we don't share every isolate that comes in. We don't notify the competent authority because that's the responsibility of the FBO to do that.
Okay, great, thank you. Definition of outbreak as used in Regulation 2005-179. EFSA defined foodborne outbreaks as incidents in which 2 or more people develop the same disease or infection following the consumption of a common contaminated food. Is it reasonable to take this interpretation when implementing the regulation?
Okay. So for any interpretation of the regulation, I invite the colleagues to contact Sante G5 because they are the one behind the correct interpretation. This is like official. So now I'm answering as Mirko, considering what is written in the FAQ that they published and taking into consideration that the new implementing regulation is an implementing act of the Article 8 of the Directive Zoonosis about foodborne outbreak, I would say yes, it's a good proxy for considering in the implementing. However, for a full correct answer and legal answer, I invite the colleague to contact G5 at some time.
Great. Okay, I think we are running out of time here, so we will leave it there, and thank you to everybody for their attention and for people who put in questions and participated in the discussion, and obviously especially thank you to our 4 speakers this morning for their time and their expertise, and I hope everybody enjoyed today's session, and we look forward to maybe you will join us in December. We will be advertising details of a kind of subsequent follow-up that looks specifically at food businesses and how they might use whole genome sequencing technology. So thank you again to our speakers this morning. We really do appreciate it, and there's lots of applause coming up on the screen that I can see there. So thank you for that, and enjoy the rest of your day.
And just to remind everyone, we will be sending out a feedback form shortly. So if you wouldn't mind filling that in for us, your feedback is very valuable. I think we might be, the second part of the whole genome sequencing webinar will be earlier than December. I think we might be end of November, Linda. So keep an eye out. Yeah, keep an eye out for the advert.
Okay,
great. Thank you, everyone. Thanks for joining us today.
All right.
Take care. Bye-bye.